Thank you everyone for joining me for my talk on the ontology-based representation and analysis of conditional vaccine immune responses using omics data. So the impetus for this project was actually a grant that we received from the National Institute of Allergies and Infectious Diseases to work on the ImmPort project. So ImmPort is an immunology data portal that combines data from many different sources, such as the Centers of Excellence for Influenza Research and Surveillance and the Human Immunology Project Consortium. All of these different organizations have a wide variety of different clinical and mechanistic studies, which you can see in the bubble diagram to the right. So we have different in some of the tests with ELISA, flow cytometry, ELISA pot, but we also have tighter results for immune response or for antibody data sets. So what we're mostly interested in is all the studies contained in this red bubble, which is focused on vaccines and specifically immunological responses or studies relating to vaccines. Namely, the fact that these data sets could be used for something we call pathway analysis. So pathway analysis is a subset of gene set enrichment analysis or gene set analysis, where you're given a set of specific genes, and then you take a look at some of the ontology or knowledge base, which maps to various other biological pathways, and you run a short statistical test to see if a specific set of genes are more likely to be associated with one pathway than the other. In this case, we have a hierarchy from the Reactome knowledge base, which shows cellular response to stimuli and immune systems. Each of these have a hierarchy which goes down to the innate immune system, which goes to neutrophil degranulation, with the color yellow being used to denote the degree of significance. So from there, we can just have a list of interesting gene sets, and then we proceed to get a better understanding of what commonalities we see, which is especially useful when we're taking a look at omics data for immune response. So the final thing that I want to bring up is that this project very much had a predecessor in the Vaccine database, which was a previous database that we worked on in focus to identify specific vaccine immune effectors. So in this case, a vaccine is any processed material that is used to generate an immune memory, and it's typically targeted against viruses or bacteria. So we found over 1,700 experimentally identified vaccine mutors when we focused on analysis of 4 specific yellow fever and influenza vaccines. And each of these pathway analysis was focused on one specific vaccine. And while this was definitely great to get a sense of what's going on, one thing that we wanted to tackle with ImmPort database is if we could perform an analysis of the multiple different types of vaccines that show up and how we would group and combine them together. So the answer to this is, of course, through the use of ontology. Namely, we used the Vaccine Ontology and a subset of Vaccine Ontology called the Vaccine Investigation Ontology in order to help make sense of the different vaccine immune response data found in ImmPort. So specifically for this paper, we had 2 teams that were working together. We have the HeLab team, which based on the University of Michigan and which I was part of, which mostly focused on the actual study metadata, making sense of how it functioned, and then later on performing some pathway analysis once the WULab, which is the Oregon Health and Science University team, focused on the actual back-end statistical program and generation of the knowledge base that we ran our pathway analysis of the ImmPort vaccine immune response data. So for the pathway enrichment analysis, all this was done for the VIGET web tool, which was developed for this purpose. So VIGET stands for the Vaccine-Induced Gene Expression Analysis Tool. And we wanted to analyze 3 distinct time points for all of our results. We have day 0, which is when a patient was being expressed the first jab of the vaccine. And then we compare their gene expression to every day 0 listing at later points at day 3, day 7, and day 14. Once we've selected all the different studies, we adjusted the analysis by all the parameters I showed you can see on the figure to the right. this is done, so you should be able to replicate our analysis on the website. From there, we have a specific gene list that we generated by specifying what our threshold is for change in log2 expression, and we included any of the genes that had a p-value of 0.05 or less. We've known from previous work that it's fine to use p-values for generating all the specific genes, so long as when we actually look at which pathways are significant, we restrict ourselves to FDR of 0.05. So once we downloaded the ImmPort database and we take a look at the actual annotations, we come up with a schema after some trial and error going through everything, where we could take a look at a specific study and then find all the different accession IDs that are stored within it. So each study contains multiple different cohorts, which are the different experimental populations that all share a common pattern for immune response. Each of these cohorts have an individual subject or person, which has an exposure extension number, which corresponds to a vaccine in this case, and also a specific sample type, which corresponds to an ELISA or sample taken from nucleated blood cells. Each of these contain a specific experimental sample, which contains the actual results, which are mapped to the GSM ID, which in turn are the expression rates for the different genes when we could finally link it back to the GEO, which is a gene expression omnibus, I believe. So we had a separate pipeline for some early ontology-based annotation before in order that was separate, but that wasn't much developed by our lab. But the main thing that we took in this context was a mapping of vaccine ontology IDs to each of the exposure extensions. So once we've created the schema of mapping these different extension numbers together, we compiled them into a nice Excel document and then mapped all the VO IDs as part of column C. So once that was done, we had at least a nice guidance for the actual Wu lab to work on VIGET and actually incorporate this data for eventual later analysis. But focusing on the vaccine ontology ID mappings and some of the statistics that we get from that. Again, I want to mention that the specific statistics are from pulled on February 28th earlier this year. We utilized an initial database download back in 2020. So we see some changes in the total listed vaccines through part of the update, but otherwise the actual framework that ImmPort used remained consistent. So pulling over 36,000 immune records, we had 6,258 that are related to vaccines. From there, we had 4,607 human subjects divided into 324 cohorts for 37 listed vaccines. So when we mapped the VOIDs and retrieved them using OntoFox, we managed to get this very simple hierarchy pulled, where you can see that we have the parent term of vaccines, and then we're divided by both the direct parents for the individual vaccines, but also all of the individual vaccines that occur as the children at the end. I will say that for the most part, A and B were existing in the prior database before that we initially looked at, while most of the COVID-19 vaccines were later additions that occurred due to the COVID-19 pandemic. So if you also count all of the children, you'll notice that there are not 37 vaccines. That's because 3 of the vaccines that we found as being different within ImmPort all were mapped to the 2008-2009 trivalent influenza vaccine. So using that diagram, we've established our first use case, which is an ontology-based query of vaccines and vaccine investigation data. So as you can see, the VO hierarchy can easily identify differences between vaccine targets. In this case, that would be the species of vaccine is targeting against, which can be found through both the clade, such as viruses, or for individual species if we're focusing on yellow fever. And of course, this query can be done through Spark or ODL, which makes it pretty easy for anyone else who wants to perform a similar type of experiment to repeat our results. However, while I showed the broad hierarchy slide before, we also imported a couple of axioms, and these axioms are used to clarify some of the additional children parents that can be inferred through a specific vaccines. And the two that I want to focus on for this talk is the idea of live attenuated vaccines and activated vaccines. Both of these are vaccines that utilize the actual virus or bacteria, which is targeted against, instead of just using a subset or the specific gene or protein. In this case, we denote both of these relationships through the has-quality relationship of either vaccine organism live attenuated, which means that the virus or bacteria is weakened but still could cause infection. And then we also have has-quality vaccine organism inactivated, which means that the virus or bacteria is either killed or otherwise unable to replicate normally. So once we've got this hierarchy, we extracted these terms, generated a JSON file that showed both the asserted hierarchy from before, but also the inferred hierarchy with live attenuated and inactivated vaccines for users to focus on. So once we've got this work, the Oregon Health Science University team focused on developing their own on VIGET, making sure that functions, and then it passed it back to me in order to perform some analysis once they've set up the pipeline to incorporate all the data using our standardization. So from literature search, we know that both males and females are known to have a differing immune response when it comes to different viruses, to different diseases, and other effects. And this, while there were some patterns when it comes to the types of response that's going on. When it comes to viruses specifically, there tended to be a fair amount of variation in the sense that you could have an influenza vaccine one year that would have worse effects on males, and then another year, the next year, it would be females who would be more severely affected by the virus. So what we wanted to see is, can VIGET identify these patterns when it comes to influenza vaccines? Now, we had already done so in the original VIGET paper for yellow fever vaccines, where we observed a specific pattern when it comes to our longitudinal analysis. But we wanted to focus on influenza vaccines, and indeed, this was a cut part of the earlier VIGET paper. So for our analysis, we found 16 studies with an ImmPort that correspond to 6 influenza vaccines. We had one single live attenuated vaccine in the form of flu mist, and then we have 5 other inactivated influenza vaccines. And from there, we took a look at the paired expression data for all these vaccines after following the standardization that I laid out earlier. One thing I want to note is that the numbers for each of the different time points varied, simply because each of the different studies only had used a different schema for when they measured experimental results, but also that some of them were inconsistent or missing individuals one. So using the smallest pairing that for the analysis, we had 210 males and 260 females. So when we completed our analysis for the combined influenza vaccines, we checked fold chains for multiple different genes and proteins at different thresholds, which you can see as part of Table 1. And we also recorded the number of genes and pathways we found to be significant for males and females. Now, for our analysis, we found that day 7 was the peak where we had the most amount of gene expression in both males and females. I still say greater amount. You'll notice that the actual count for significant pathways is heavily skewed towards females, while males only having more significant genes during some of the higher thresholds for day 7. That doesn't actually hold true when we actually take a look at the significant pathways to make sense of what's going on. We also noticed that in terms of peak, that females tend to have a much higher number at day 3. It increases a little bit during day 7, not nearly as dramatic as for males, before dropping down to about the same level on day 14. Meanwhile, males have their peak at day 7 and drop down day 14. But there seems to be some sort of temporal lag when we take a look at which pathways are found to be significant. So this is nice as a summary figure. What does that look like when we map it to reactome? And it looks like this. So we mostly focused on cellular response to stimuli and immune system, as those were the two pathways which had most of the details. And the big thing you can see is that females are showing significant pathways as for the immune system during the first week, while males only have neutrophil degranulation as being the most obvious term in day 7 before kicking up a little at day 14, where we have more general immune system response. We also see that males are starting to have cellular response to stimuli in day 14, while that to a lesser extent is going on in females in all 3 days. So altogether, this is getting a sense that for males, they're not really having much reaction or to most of the immune system until after 2 weeks have passed, while females seem to be responding within the first 3 days. So again, I want to point out some of the significant pathways that we found for males. I mostly listed children of specific terms because I didn't want to list out immune system, innate immune system, etc. for females. And while we both see neutrophil degranulation as popping up, as being significant, more so for females than males. We're also seeing some other interleukin-4 and 13 signaling, interleukin-10 signaling, and an inflammasome pathway response. However, separate to all the immune response, we also quite clearly found that females have a specific relation for cellular response to stress. And while the previous figure was shown just using a magnitude of change, we actually went back and took a look at patterns for what happened when we take a look at only genes that are have a greater amount of expression later on versus those that were down expressed. And we found that all of the genes related to cellular response to stress in females, they were up, which surprised us and made us want to follow up, which I'll do so for the discussion. So once we've done that, question we wanted to ask is, okay, when we combined all of our different vaccines together, we get some really interesting and useful results. But the next question is that what happens when we try to narrow down to a smaller subsection when using all these different studies? Will we be able to find something more specific or not? And that, if any of you can guess, led to a separate issue, which is that because we're cutting down on a cross-section of results, that meant our pairings from the number of genes are getting smaller and smaller, which affected our analysis. One of the big ones is that the day 0 to day 3 female live attenuated influenza vaccine analysis would not run on VIGET, simply because the data size was way too small. We had a bit more luck with inactivated vaccines, simply because that was 5 out of the 6 vaccines we performed analysis on. And even though we found some terms being a bit more enriched, again with general immune systems, it's still much less power than we had before. The main thing that I did want to highlight is that while males are having a different immune system pathway, is that females were starting to see programmed cellular death as another term that's being enriched at this day 14 point, which I'll get to, which combined with what we saw with cellular response to stress paints an interesting picture of what's going on to females when it comes to the influenza vaccine. So circling around back to the applications of ontologies to ImmPort studies, our study successfully used ontology in order to guide the analysis of ImmPort. In this case, we had VIGET successfully used for influenza for this study and for yellow fever vaccines as part of the initial paper that published earlier this year. Again, the main difficulties we had with this mapping was just making sense of how ImmPort structured the data, which the main thing that I remember is a controversy over the definition of day 0, because there were some studies that use the date of vaccine administration as day 0, while other studies use the day of enrollment into a study as day 0. And that has a significant effect if you're trying to take a look at the actual differential gene expression response, because we assume that our start point was right before you get the vaccine or it has an effect. Now, while this was a hassle to understand, ImmPort fortunately had a lot of tables that list out the different structure for their database, which, while it was lacking very good descriptions and required you to go through each database to figure out what's going on, it was still well annotated enough that this could be replicated and eventually made sense of. So once again, I want to, either using VIGET or using a different set of studies, the same framework could be used to analyze diseases or through utilize different procedures in vaccination routes, for example, either going through the nose or through the muscles, or also differences when it comes to patient phenotypes beyond sex. So while ethnicity would probably be one of the easier ones, I still want to give a word of caution when trying to analyze using age, mainly because ImmPort stores age as a series of range at the cohort level instead of the actual subject level, which means that for any specific data set, we only know that it was taken from a person potentially from 28 to 65 years of age, which again causes some complications and which restricts how useful it actually is for analysis, unless you have access to the original data sets with the ages listed. So circling back onto the actual effects of biological sex for vaccine immune response, we saw a fairly consistent pattern of males having a delayed differential gene expression response to the different vaccines. So with it really starting to ramp up after 2 weeks, while females are showing both a stronger response earlier on, they're also showing a stronger pattern when it comes to cellular response to stress. Now, when we were doing some further literature search to make sense of the situation, especially when it came to neutrophil degranulation, we found that neutrophil degranulation was linked to various inflammatory diseases or other autoimmune disorders, and that the actual apoptosis that we saw on day 14 was also a symptom of excess cellular stress. So all of this is gesturing to me that, at least for these influenza vaccines, that there is some sort of effect or cause that's causing females to react worse when it comes to the actual adverse events or just their body feeling a bit down within the initial weeks. Now, the reason why I suggest that is that we're quite uncertain on the exact cause for what's going on. So literature indicates that females tend to have a stronger immune response to things in general and more biased to autoimmune disorders or just having a stronger stimuli for what's going on. While males are more likely to have issues with diseases that caused by suppression of the infective immune response. So we're uncertain if that is just an innate feature that we should expect for any source or antigen, or if it's also a case specifically in the formulation and how we test vaccines, in that all these vaccines that have been approved and go through clinical trials are willing to err on the caution of, in order to be efficacious in the general population, we're willing to accept slightly higher or somewhat higher cellular response to stress when it comes to females. So that's an open question that we would like to tackle, but that's something we haven't done yet. So anyway, I just want to acknowledge the funding for this project, and again, the help of the Wu Lab at Oregon Health Science University for development of VIGET and enabling me to run the tools to perform this analysis. Thank you.